Good morning, good afternoon, and good evening, everyone, and welcome to the 9th webinar in the Cardiovascular Connections 2022 series. Today's webinar is titled Cardiovascular Regenerative Medicine, Deconstructing Regenerative Therapeutics, featuring Dr. Eduardo Marbán, Executive Director of the SMID Heart Institute at Cedars-Sinai Medical Center. Today, he'll dive into the complexities of the regenerative medicine, cardiovascular medicine, and the future directions for cell therapies. Before we get started, we'd just like to acknowledge our partners at the American Physiological Society and the European Council for Cardiovascular Research, and also to thank the sponsors of this webinar series for making it all possible. And now I'm pleased to welcome Dr. Marbán to the floor. Dr. Marbán, take it away whenever you're ready.
Hello, everybody. Today I'm going to tell you a story that goes back really 22 years when we started working with embryonic stem cells, seeking to differentiate them into beating cardiac clusters for use as potential biological pacemakers. That work led to some proof of concept studies in vitro and in vivo, but we became somewhat disillusioned because of the persistent immune problems as well as the arrhythmogenic properties of the current generation and previous generations of pluripotent stem cell derived cardiomyocytes. So we scratched our heads for a while, and then 18 years ago dedicated an effort to use the adult heart as a source of cells, thinking that perhaps there would be some rare cell type within the heart that was dedicated to promoting healing and tilting the balance away from fibrosis towards repair and recovery. And if we were able to isolate such cells and put them back in, that we might be able to tilt the balance in that direction. So it's that journey that began with a really pretty simple-minded idea, and that is, let's find a regenerative cell type, transplant it into the heart, into regions of injury, let those cells engraft, proliferate, and differentiate, and create new heart muscle. In other words, a canonical stem cell mechanism that, in a way that we couldn't have foreseen, led to today's work that I'll culminate in, and that is the creation and validation of new non-coding RNA drugs for a variety of inflammatory and fibrotic diseases. So just to reiterate, our initial goal with cell therapy 18 years ago was to repair the permanently injured heart, recognizing the dogma that fibrosis in the heart once formed is irreversible, and that cardiomyocytes once lost will never be recovered. But our updated goals after 18 years of discovery work are as follows, and that is to use cells, exosomes, or novel RNA drugs as therapeutic candidates for a broad range of inflammatory fibrotic diseases, in no way limited to the heart. So in a fantasy version of today's talk, I would talk about the discovery of the cell type of interest here, CDCs, and then argue that through a set of predictable and iterative steps, this led to the development of the novel RNA drugs, which are now the focus of much of our lab's attention. But in reality, it was a circuitous path noted by various inflections. All of the inflections were driven by surprising scientific insights. And had it not been nonlinear like this, it wouldn't have been nearly as interesting. As I go through the talk, you'll see the squiggle recur on the lower left-hand corner at points of inflection where some aspect of the data forced us to rethink the paradigm and move on to the next logical step. So the first step in all this was to start with human hearts and develop a protocol for isolating what we believed to be human heart progenitor cells. And the protocol is shown in the schematic at the top. We started with cardiac tissue, human cardiac tissue, which we cut up into small bits about the size of a coarse grain of sand called explants. And these were in primary culture. And spontaneously, under fairly routine culture conditions, these explants yield over days to weeks, a lawn of cells that cover the bottom of the dish, and these are called explant-derived cells. Those cells can be scraped off and replated in dishes that repel adhesion by virtue of being coated with polylysine. And under those conditions in primary culture, the cells self-assemble into little organoids called cardiospheres. And this process was first described from human hearts by Elisa Messina and colleagues at Sapienza University in Rome. What we did, working initially with Elisa, was to take cardiospheres and replate them on adherent media and passage them for several passages to create the cell product of interest, cardiosphere-derived cells, or CDCs. And so what are CDCs? Well, in this time capsule of knowledge that goes back to 2004 to 2007, we recognized that these cells were from the human heart, and they were progenerative cells in the sense that they could differentiate into multiple cardiac lineages with some alacrity in vitro and with some difficulty in vivo. But we were able to find needle and haystack events histologically when we implanted human CDCs into skid mice in the heart of instances in which they became cardiomyocytes, smooth muscle cells, and endothelial cells. We recognized that these cells were antigenically unique. They weren't exactly like mesenchymal stem cells or any other described cell type. They're CD105 positive uniformly. That's endoglin, and they're uniformly negative for CD45 and other markers of hematogenous origin. They also tend to be low CD90, which differentiates them from MSCs. So thinking in terms of the canonical stem cell mechanism of transplantation, engraftment, and differentiation, we focused on an autologous paradigm whereby we used a biotome to harvest small bits of endocardial tissue from subjects who had undergone a myocardial infarction and were left with mild heart failure with reduced ejection fraction, abbreviated here as HFrEF. We studied 25 patients, and the patients who underwent biopsy had their cells, their heart tissue converted into CDCs, and some weeks later, after the biopsy, had a reimplantation procedure involving a catheterization down the infarct-related artery and intravascular delivery into the zone of the MI. The control group was standard of care. This was a 25-patient study, and we showed some evidence of regeneration as well as functional improvement. And of course, the underlying dogma was that the mechanism of action was a canonical stem cell mechanism. The initial work was published 10 years ago in The Lancet, and that was a 6-month follow-up, and then the 12-month follow-up was published in the Journal of the American College of Cardiology, and there we focused on things like regional measurements of function by cardiac magnetic resonance imaging, and we found if we compared peak strain within the tissue, the more negative, the more contractile the region is, and this is in the region of the infarction, the left-hand panel. The CDC subjects had a more negative strain, indicating higher regional contractility. Systolic thickening, which is obviously a direct indication of how well the heart is working, was significantly greater in the CDC group, and likewise thickness was greater, signifying that there had been some correction structurally, and these parameters were not significantly different between the two groups at baseline. So we found these data to be somewhat encouraging, and we were thinking along the lines of what to do next, but here came the first sort of moment of surprise, in the sense that we ended up questioning the mechanistic rationale for autologous therapy, which is indicated here. And that would be the idea that if you transplanted CDCs into the heart, the cells would proliferate, differentiate into multicellular new myocardium of donor origin. But instead, even while the Caduceus trial was taking place, we were making progress preclinically, and we demonstrated using a variety of means that the cells instead would only engraft for short term in the heart, whether they were syngeneic or allogeneic. In other words, whether they were a perfect genetic match or some other strain of rat or pig, let's say. And that led to new healthy tissue of host origin, which by inference means that there must have been something secreted by the cells while they were in the tissue that stimulated the host tissue to proliferate and heal. And the data for this is in the citations below. And in studying this process, we realized that this paracrine effect was mediated by something we ended up discovering to be exosomes. At the time, we thought were cytokines. But this led to a number of effects within the tissue that were not necessarily directly regenerative, but all of which were salutary, including anti-inflammatory, immunomodulatory effects, and meanwhile the cells were evanescent. So the first step in deconstructing the data, deconstructing regenerative medicine and following the data, was to go from the autologous therapy paradigm to a recognition of durable benefits despite cell transience in the heart, leading to an allogeneic paradigm. Why put individual subjects through the risk and misery of having their endocardium sampled with a biotome under invasive conditions in a hospital when we could just use off-the-shelf therapy from donor hearts? And that allogeneic paradigm has persisted for the decade since and has led to a number of clinical trials. And this realization, as you can see in the bottom left, was the first major inflection point. And so then going back to 2012, this was our updated understanding of CDCs and their mechanism of action. Even though they did qualify as cardiac progenitor cells or cardiovascular progenitor cells, given the fact that they were clonally derivable and that they were capable of multilineage differentiation, we did not have any evidence really that they worked that way in vivo. And so we rethought of them now as cardiac stromal cells or stromal/progenitor cells. And we recognized that they worked not by direct canonical mechanisms, but by secreting things. And among the things that we implicated at the time were SDF1. That led to a number of allogeneic trials that I won't discuss, including All-Star and Dynamic, both of which showed evidence of disease-modifying activity. All-Star was a phase 1 and phase 2b study of allogeneic CDCs given down the infarct-related artery after myocardial infarction in patients with mild HFrEF. Same patient population essentially as Caduceus, but now taking off-the-shelf therapy. And this led to evidence at 6 months and at 12 months of functional and structural improvement in the patients who had received CDCs relative to placebo in this double-blind placebo-controlled trial. Dynamic was a much smaller study in patients with advanced heart failure with reduced ejection fraction. And Dynamic was the first to use multi-catheter non-occlusive invasion to achieve complete intracoronary coverage of the myocardium. But today I'm going to focus on the program that has legs and that has promised to go ahead and be continued into the clinic, and that is the Duchenne muscular dystrophy program. But before I get into that, our understanding, just to reiterate, was that the mechanism of action was not canonical. Instead, it was paracrine. And among the benefits of the paracrine factors were promotion of cardiomyogenesis, prevention of cardiomyocyte apoptosis, and the recognition that the effects were anti-fibrotic and anti-inflammatory. And those two last things will persist for the rest of the talk as foci of mechanistic work that's led to these non-coding RNA drugs. So the HOPE Duchenne trial studied, and HOPE stands for Halt Cardiomyopathy Progression in Duchenne, focused on Duchenne muscular dystrophy, which is a rare X-linked recessive disorder. It's rare, but it's the most common musculoskeletal crippling disease in the world, and often leads to disability and death by the first and second decades of life. The most prominent manifestation is a skeletal muscle disease, and this is often manifest for the first time when boys who are affected with Duchenne struggle to get up from a lying down position without using their arms. And this becomes so prominent that the parents are concerned, take the child to the pediatrician. Pediatrician does genetic testing, and that confirms the absence of the dystrophin protein, which is the molecular cause for Duchenne. And at that point, the child's prognosis goes from totally normal to dismal, and the parents hear the words Duchenne muscular dystrophy, which they'll sadly never ever forget, and it will change their lives forever. So what's less appreciated in the popular conception of muscular dystrophy is that the heart is affected as well. And over time, this leads to progressive replacement of living heart muscle by scar, which can be seen here by gadolinium enhancement as white areas within the heart after the tracer is infused. And a normally dark myocardium turns to white in areas of hyperenhancement. The pathogenesis of Duchenne is, as I've mentioned, one of dystrophin deficiency. Dystrophin being a massive, long protein that links the extracellular matrix to the cytoskeleton, and in so doing creates a macromolecular complex. All of that falls apart in the absence of dystrophin, and membranes become leaky, and which, when you think about it from first principles, if membranes are susceptible to wear and tear, that's going to be manifest most likely in contractile tissues that are forcing the cell membrane to undergo stress on a beat-to-beat basis in the case of the heart, or during any kind of activity involving skeletal muscle. So this leads to cell membrane damage, leakiness, and increases in cellular calcium, and a number of processes notable for oxidative stress, inflammation, and fibrosis, all of which are in principle amenable to what we knew at that point were the mechanisms of action of cardiosphere-derived cells. So we initiated the HOPE Duchenne trial, and it was motivated largely by data such as these in patients looking at the natural history of progression of cardiac scar in Duchenne muscular dystrophy patients. DMD patients who are followed in a clinic, typically now for prognostic purposes, get a cardiac MRI yearly. And when that is done, the first cardiac MRI that reveals any portion of the heart involved in scar is a prognostic indicator. On the left here, you see on the y-axis something that says LGE positive LV segments, and basically this takes into account how many segments of the heart, using an AHA segmentation model of 16 total segments, how many of those are evidently involved in scar. And on the x-axis here is the age of the patient at the time of the study. And what you can see in each line, connected line, is one patient. So you can see that as soon as you get one segment is involved in scar, next time often there's 2 or 3 or 6 segments, and there's progressive conversion of living myocardium to fibrotic tissue. So it's a progressive, irreversible disease culminating in death. So we decided to study CDCs in a mouse model called MDX, which is a naturally occurring model of Duchenne muscular dystrophy that arises from a mutation that stops the translation of the transcript prematurely, and the mutation is in exon 23 of the mouse dystrophin gene. So these animals get a skeletal myopathy that's evident as soon as they're weaned, and a cardiomyopathy that's evident later in life, but certainly quite evident by the 10th month, which is where we start here at baseline on the left. And we're tracking here animals over time, baseline being 10-month-old MDX animals, and we're doing serial echoes to quantify left ventricular ejection fraction, which is what is plotted on the y-axis. And what we did at baseline after phenotyping is we opened the chest and injected CDCs into the myocardium, and we randomized the groups so that some of the animals received CDCs plus vehicle, and other animals received only vehicle. At first, both groups had the same ejection fraction, but you can see that over time, the first point being 3 weeks, and the second one being 2 months, and the third being 3 months after injection, we begin to see the curves bifurcate. The ejection fraction is improved or sustained in the group that got CDCs, but declines progressively over time in the group that was injected with vehicle. At month 3, after phenotyping, we opened the chest again, injected CDCs or vehicle again, and continued to follow up. And you can see that for another 3 months, the same benefit was maintained, so that EF was boosted or sustained, while in the control group, the ejection fraction continued to fall. So the control group is following the natural history of progression of cardiomyopathy in these animals, whereas the CDC group shows sustained functional benefit. What was somewhat surprising, though, is that in a separate group of animals, we measured treadmill distance. And an animal, a normal wild-type control of the same strain under these conditions could run about 700 meters before getting exhausted. But an MDX mouse of this age could only run about 250 meters, and that's plotted on the y-axis here. And after baseline treadmill testing, we then did the operation to inject either CDCs or vehicles into the heart, and then followed treadmill time weekly. And you can see that as soon as 3 weeks post injection and sustained for all 12 weeks of follow-up, there was a significant increase in treadmill time, treadmill exercise tolerance in the animals that had received CDCs relative to vehicle. This was very notable to us because this change from about 250 to over 400 was not only huge, but also unlikely to be due to the level of cardiac dysfunction that we saw in these mice. The normal ejection fraction in a mouse is about 75%, and yes, these animals begin with an EF of about 60%, but they're hardly in class 3 or class 4 heart failure. So we suspected that this was a skeletal muscle effect, which we verified by taking isolated skeletal muscle and measuring its force in a dish. And indeed, in the animals that had received intracardiac CDCs, nowhere near the skeletal muscle, the isolated skeletal muscle measured weeks later was hypercontractile relative to those that had received vehicle. And by this point, we were recognizing the existence of circulating factors and had some idea of what they were. So we went on with that insight to put skeletal muscle function as one of the endpoints in the HOPE Duchenne trial, even though we were targeting the heart. So as I told you before, HOPE stands for Halt Cardiomyopathy Progression in Duchenne, and we enrolled Duchenne muscular dystrophy patients who were at least 12 years old, and at baseline had at least 4 segments of their hearts involved by scar by cardiac MRI. We enrolled 25 patients in a 1-to-1 randomization scheme, and the randomization was basically standard of care, which included steroids in all patients, or multivessel infusion of allogeneic CDCs. And then the follow-up was the same in all the patients, and really focused on safety, on scar by MRI, and also cardiac performance by MRI, and as well as performance of the upper limb, or PUL, which is a rigorously quantifiable measure of how well the arms are working. And why did we focus on the upper extremities? At enrollment, if we stipulate that at least 4 segments of the heart would be involved in scar by MRI, and the patients be at least 12 years old, almost all these patients were already wheelchair dependent. And that makes them really quite dependent on arm function for any measure of independence. They need to use their arms and hands and shoulders and elbows for things like lifting a drink to their lips, for hugging their moms, for using a joystick or a mouse. And the joystick is often the way they manage to achieve ambulation using a wheelchair. So we felt this was a very meaningful functional measure in a population that's been largely ignored in all previous clinical trials of Duchenne, because the dogma is that once they're wheelchair bound, these poor individuals have no hope of palliation or improvement. So here's the findings of HOPE at 6 and at 12 months. CAP 1002 here is the name that the company that sponsored the trial, Capricor, gave to the allogeneic CDC product. And what you can see in the upper left panels are the fact that at baseline, both the subject that's been treated with CAP 1002 or is being treated with CAP 1002 and the usual care subject have significant bright spots within their hearts, signifying that they both have a significant scar burden. But in the CAP 1002 subject, when you compare the 6-month and the 12-month images relative to baseline, there's some diminution of that bright spot density or frequency or how much of it occupies the heart, whereas in the usual care subject, that white stripe on the free wall and posteriorly seems to be quite prominent and undiminished. And in fact, the statistical analysis of the pooled data on the bottom left showed that that was a significant difference over time. When we measured the performance of the upper limb, we found, and as you can see in the lower right in particular, that there were many more patients whose performance of the upper limb was either sustained or improved in the CAP 1002 group than in the usual care group. So we were encouraged by these findings, but also recognized that this was even though it was randomized and prospective, it wasn't double-blinded or placebo-controlled. So we were encouraged by these data, but not totally convinced. Meanwhile, we had discovered in another inflection point in our journey, as shown in the lower left corner of the squiggle and the symbol of a person moving around this maze, that exosomes, or a category of extracellular vesicles, were the key mediators of the benefits of CDCs. So exosomes, as indicated in the left-hand panel, are manufactured by a very specific cellular pathway that involves invagination of the surface membrane as endosomes that then fuse with products of the Golgi to form multivesicular bodies. And that process is dependent on neutral sphingomyelinase, abbreviated here as NSMASE, and that is a part of the ceramide biosynthesis pathway. Multivesicular bodies are either targeted to lysosomes where they're degraded, or they fuse with the surface membrane and are secreted, the contents are secreted as extracellular vesicles. Exosomes tend to be tiny. They have a modal diameter of about 100 nanometers, which is the same as an HIV-1 virion, just to put it in context. Why did we believe that exosomes were important for CDC actions? Those data, which first appeared in a paper by Ahmed Ibrahim from my lab in 2014 in Stem Cell Reports, but are abstracted here, are on the upper right. We are looking at the progress of left ventricular ejection fraction over time after a massive anterior myocardial infarction in mice that received one of two injections. Either their hearts were injected, and this is shown in the green line with CDCs, or they were injected, as shown in the blue line, with CDCs that had been exposed previously to a small molecule GW4869 that's an inhibitor of NSMase in that biosynthesis pathway. They were not secreting any exosomes, but they were apparently healthy in terms of membrane integrity and metabolism. Injection of CDCs has a terrific benefit and improves EF over time, whereas injection of CDCs that had been exposed to GW4869 has no benefit at all over time. That to us meant that exosome secretion is necessary for CDC benefits. Now, in the lower right-hand panel, we look conversely at taking the cells out of the equation entirely, and now what we're injecting are exosomes that have been purified from media conditioned by CDCs, but the only biological material we're injecting are the things that are pulled down when we isolate the vesicles, including the vesicles themselves, of course. So here, the green line, again, in a population of mice that have undergone a large anterior myocardial infarction. The green line shows the trophic benefit over time of having CDC exosomes injected into the heart, similar, if not a little better, than what we had seen with CDCs themselves. But the blue line here shows what happens when you inject only vehicle. Now, what is the red line? It actually shows that this effect of exosome injection is quite specific to the cell type of origin, insofar as skin fibroblasts, which also secrete exosomes with alacrity, if you isolate those exosomes from media conditioned by fibroblasts and inject that instead, it has no benefit over time in terms of functional improvement or structural improvement. So top right indicates that exosome secretion is necessary, and bottom right indicates that exosomes are sufficient for CDC benefits. Then we realized, and this was a logical extension of the idea that cells secrete exosomes. Exosomes are known to be able to go throughout the entire body via the circulation, no matter where they're secreted, and recognizing also that injection of CDCs into the heart has remote effects on skeletal muscle in the leg and on skeletal muscle in general. We did the next logical experiment, and that is to test whether intravenous CDCs work just as well as intracardiac CDCs. We tested this again in MDX mice, and as you can see in the top left, the protocol was simple, and it just involved randomizing MDX mice to intravenous injection via the tail vein of CDCs and follow-up for 21 days, or intravenous vehicle with follow-up for 21 days. In the top right, the ejection fraction when comparing baseline to 3 weeks was improved in the CDC group, but not in the vehicle group. Likewise, exercise capacity. And on the bottom right, we also showed that cardiac fibrosis was markedly reduced in the CDC group relative to vehicle. So this inspired us to attempt intravenous cell delivery in humans, which was yet another inflection point. And that led to a follow-on trial of CDCs in Duchenne patients known as HOPE-2. Now, HOPE-2 was a properly designed, very rigorous trial protocol in which we randomized patients double-blind in a one-to-one fashion again. It's a double-blind study. Neither the patients nor their treating team knew what had been infused, and this was placebo-controlled. The bottles were masked and looked identical, and the subjects were very similar to those enrolled in HOPE. They had advanced Duchenne muscular dystrophy and already substantially reduced skeletal muscle function and cardiac function when they were enrolled. The primary objective was to evaluate the safety and the efficacy of this allogeneic formulation of CDC's CAP1002. And what we decided to infuse was 150 million CDCs intravenously every 3 months. And the fact of intravenous delivery was motivated by the previous work in MDX mice that I've shown you, and this work, and the fact of every 3-month delivery was motivated by our previous finding that multiple injections led to sustained benefit in MDX mice. 9 sites, all of which were specialized Duchenne muscular dystrophy centers, were involved. The population under study comprised 20 subjects, which is not unusually small for a very rare disease. The patients were 14 years old on average. All of them had to be on corticosteroids as a condition of admission into the trial. Corticosteroids having been the only pharmaceutical to be approved to slow the progression of Duchenne muscular dystrophy, we wanted all patients to be on it to eliminate any potential confounding effects of that as an independent variable. And at the time of enrollment, 80% of the patients were already non-ambulant. Here are the data published recently in The Lancet. First, I'm showing you here the trajectory of performance of the upper limb as basically as a deviation from baseline in 2 populations. In blue, the subjects who received intravenous CAP1002 as compared to the trajectory over time of the subjects who had been randomized to and received 4 doses of placebo. And you can see a broad separation between the two lines. Is evident very clearly by 6 months and even greater at 12 months. And the statisticians concluded that this was equivalent to a 71% slowing of the progression of loss of arm function over time, which is very clinically significant. And if you look at the bottom line in that sort of plot of a number of confidence intervals for individual measures of arm function, the global statistical test of the pull measures at the bottom had a significance level 0.0087 when comparing the 2 groups. A very strong effect that, despite the small size of the study, indicated the consistency of this benefit. For this audience, of course, the cardiac findings may be of even greater interest, and so the panel just revealed shows left ventricular ejection fraction by MRI over time. And you can see here in the red that the patients randomized to placebo follow the natural history of ever progressive decline in EF over time, and the patients who had been randomized to CDCs had total preservation, if not slight improvement of function over the 1 year of follow-up. And here, the global statistical test of cardiac measures, which is now highlighted with a red box, showed a significance level between the 2 groups of p equals 0.0066, extremely significant and very encouraging data. So we now update the understanding of CDCs as follows. The methods and the bioactivity of these cells have been reproduced by more than 75 labs worldwide. And we now know that the characteristics include secretion of exosomes. And we are now moving on. And in fact, HOPE-3, which is a larger version of HOPE-2, with 68 patients, also one-to-one randomization and placebo-controlled, has been started, and that trial, if even successful to the extent that the effect size is even half of that, what was seen in HOPE-2, will be considered a home run, and FDA has signaled that this product may be approvable for Duchenne muscular dystrophy on that basis. So in a field that has admittedly been fraught and full of controversy and missteps and retractions and a lot of hype, this may be the last cell product standing and maybe the first to progress all the way through to commercial approval for a cardiac or mixed cardiac indication. So the next step then, of course, once we understand that exosomes are the mediators of benefit, is to figure out what is it within the exosomes that is actually producing the benefit. And we started that journey in 2014 and first focused on microRNAs within the exosomes because we knew what microRNAs did. And so we performed microRNA-focused arrays on exosome contents and found that they included these exosomes from CDCs, included 146A, top right, and did some of the logical experiments to show that 146A was contributing to the benefits. And then in a different set of experiments, we also looked at 181B. And even though we and the rest of the people were kind of excited by the microRNA stuff, we had some cold water thrown on that theory when we actually looked at in an unbiased way, not microarray focused, not mirror focused microarrays on the exosomes, but rather next generation RNA sequencing of all the RNAs. We found that on the left pie chart, which is the content of CDC exosomes, the bright blue slice with a 7.07 indicates that only 7% of the small RNAs within extracellular vesicles were microRNAs. And so we thought, well, I mean, these are there, but are they numerically plentiful enough to really play a role? And this forced us to think, and was yet another inflection point as indicated on the squiggle diagram on the bottom. And so we ended up refocusing on slices of the pie that were other kinds of RNA that we didn't understand nearly as well in terms of potential mediators of disease-modifying bioactivity. And among the pieces that really caught our eye were the sort of bright green slice, which consisted of about 19% of the pie. And the reason that we particularly focused on that segment, which consists of something called yRNA, is that the species within that yRNA pie, as I'll show you in the next slide, were exceptionally plentiful as individual species. I should say, when you do unfocused RNA sequencing of extracellular vesicle contents, such as we did for CDC exosomes, individually, some 30,000 RNA species are identified. A bewildering variety that includes not only yRNAs, but also mirs, long non-coding RNAs, piRNAs, SNO-RNAs, and things that we have yet to categorize. So we focused on that yRNA slice of the pie, and the right-hand side shows that all of these brightly colored parts of the pie are much less plentiful in exosomes from normal human dermal fibroblasts that really contain mostly transfer RNA, and that's the brown segment. And so this forced us to think maybe we were being misled by the fact that we understood mirs. They weren't very plentiful. Instead, we focused on, and we identified one particular yRNA called EVYF1, extracellular vesicle derived Y fragment 1. And the reason that we did is indicated in that yRNA abundance chart on the top left. This yRNA species, EVYF1, was present in 350 plus thousand copies in reads in a library, in the next generation sequencing library, where a typical mirror was present in only 1,000 or so copies. And so what is this thing? Well, it's, first of all, it aligns perfectly. It's a 56 nucleotide fragment. It aligns perfectly, and I call it a fragment because it is the 5' 56 nucleotides of the human yRNA4 gene as revealed by the alignment in the top right. What is the human yRNA4 gene? Well, first of all, there are 4 yRNA genes in the human genome and 30-odd pseudogenes. yRNAs are known as partners for ribonuclear proteins Rho and LA, which are circulating antigens in lupus patients and in patients with other autoimmune diseases. In fact, that's how the ribonuclear proteins and their binding partners of yRNAs were first discovered some 40 years ago. The complex of yRNA with Rho and LA has effects on DNA replication, but there were no known benefits of any of these fragments or any disease-modifying bioactivity. But we found this fragment to be too prominent to ignore, so we synthesized it and put this 56 nucleotide RNA in a transfection reagent and infused it intravenously, where it reduced infarct size, and it's in that citation on the bottom by Linda Cambier from the lab. But also shown here in the lower right are the effects of this synthetic yRNA fragment, EVYF1, in a transfection reagent on bone marrow-derived macrophages. And you can see on the left set of bar graphs that the transcript levels of IL-10, an anti-inflammatory cytokine within macrophages, were boosted almost 20-fold by exposure to EVYF1. And so was the level of secretion of IL-10 in the media, shown in the lower right. So we thought, okay, well, this is interesting. Here's a naturally occurring non-coding RNA of unknown function that we might have just thought was junk, and it turns on IL-10 and decreases infarct size. But should we make this into a drug? And so we started scratching our heads and basically realized, well, it's interesting. It's 56 nucleotides and it works, but it doesn't conform to the general properties of FDA-approved non-coding RNA drugs. There are 12 non-coding RNA drugs that are approved by collectively either by FDA or by EMA, the European Medicines Agency. And they all share some chemical properties. They're all between 18 and 30 nucleotides long, so they tend to be short. And they're all chemically modified to improve potency and stability on the one hand, and also to decrease immunogenicity. RNA tends to elicit strong innate immune reactions when injected into mammals. And by chemically modifying the compounds, you can decrease that immunogenic reaction. It also turns out that all of the known and approved non-coding RNA drugs are designed specifically with a target in mind to suppress some disease gene in particular. So there are things like small interfering RNAs or aptamers, and a lot of those are exemplified here in the chart. But we wanted to conform to the chemical properties, so basically to have a pathway ahead for clinical translation and development. But we recognized that unlike these things that had known mechanisms and molecular targets, we were dealing with something that we discovered working backwards, and now we had to work further backwards to understand the mechanism. So we started with EVYF1. It's 56 nucleotides, focused in on a region that we'd called NT4, which was in the early 5' region, the 24 nucleotides, and then further modified it by introducing chemical modifications known as locked nucleic acids to stabilize ribose bridges and introduced a point mutation so the sequence was no longer a natural one, and came up with the therapeutic candidate TY1, therapeutic yRNA1, which will be the focus of the rest of the talk, and that was a major inflection point for us as well, as indicated in the lower left. So why are we excited about TY1? Well, it has massively interesting effects on gene expression in macrophages, on the heat maps as shown on the left, and on the fact that anti-inflammatory genes are consistently upregulated and pro-inflammatory genes are consistently downregulated, as are genes involved in fibrosis, downregulated, and in hypertrophy. So TY1 then is a bio-inspired. It's bio-inspired by having had this uncle in EVYF1, but it's now completely different, 24 nucleotides, mutant, liquid, locked nucleic acid modified, new chemical entity, which suppresses inflammation, fibrosis, and hypertrophy. It's not targeted, unlike siRNAs or aptamers. We figured out that the mechanism was not that of associative canonical yRNAs, which involves binding with Rho or La, because in TY1, the Rho 60 binding site is completely disrupted and the La binding site is absent. And then we further confirmed that this is not the mechanism because biotinylated TY1, when used to probe a cell lysate, doesn't pull down either Rho or La. But it does pull down a nuclear basket protein, TPR, which is part of the nuclear pore complex. And that nuclear pore complex is a very intimate and required shuttle for messenger RNA coming out of the nucleus, leading to the working hypothesis that we have on the lower left here, that a TY1 nuclear protein interaction alters mRNA transport to decrease inflammation, fibrosis, and hypertrophy. So we decided to test whether this works in a mouse model of heart failure preserved ejection fraction, where inflammation, fibrosis, and hypertrophy are all quite prominent. The top left shows the protocol of implementation of the two-hit mouse model of HFpEF that's recently been popularized. This involves feeding the animals L-NAME to cause hypertension, as well as a high-fat diet. The phenotype is evident by 5 weeks, at which time the animals are randomized to receive intravenous TY1 in various dosing regimens or vehicle. And we quantify things like top right at the end after 10 weeks of treatment, E over E prime, which is an echocardiographic indicator of diastolic function. The higher that number, the worse the diastolic function. And you can see a big difference between control mice that weren't fed these diets, normals in other words, and the animals that got vehicle, way up in terms of this EE prime. But any of the dosage groups of TY1, the ones that got it once every 2 weeks, once a week, or twice a week, largely corrected E over E prime. Bottom left, treadmill distance, you can see that that's much decreased in vehicle relative to control, but at least the once weekly and twice weekly doses of TY1 correct that. Circulating levels of BNP, a biomarker for heart failure, also corrected by any dose of TY1. And astronomically high circulating levels of the pro-inflammatory cytokine IL-6 in vehicle were also brought back down to normal levels by TY1 infusion. Very encouraging, and also consistent with the fact that when we measured within the myocardium families of genes related to senescence, fibrosis, and inflammation, there was coordinate upregulation in disease and downregulation with TY1. So what about other diseases of fibrosis and inflammation? Duchenne, of course, is one. Scleroderma might be another. And so we implemented the MDX model of Duchenne muscular dystrophy, and you can see in panel B that after 8 weeks of treatment, starting again with animals that were 10 months old and already had a cardiomyopathy, ejection fraction was higher in the TY1 group relative to vehicle. The treadmill distance was increased, and these parameters were all equivalent at baseline. Isolated muscle torque, meaning isolated muscle force during tetanic contractions, was more than double in the TY1 group, and fibrosis was consistently decreased both in the heart and the tibialis anterior muscle. So TY1 given after Duchenne is already established improves EF, exercise tolerance, muscle force, and decreases fibrosis. Finally, we sought to treat a non-cardiovascular cardiac, or at least a disease that only has some cardiac involvement, but predominantly is a systemic autoimmune disease, Scleroderma, that affects mostly women of middle age. This is a horrible disease that has a 29% 10-year mortality, and not only affects the skin, but often the heart and lungs, and systemic sclerosis. And this is an absolutely incurable disease with no specific drugs or therapies. So we implemented a model of this disease in mice with intradermal injection of bleomycin, which led to skin thickening. This was corrected by intravenous infusion of TY1, but not of a scrambled version of TY1 in a transfection reagent as shown in the label called SCR. Of greater interest to this audience, it also decreased cardiac fibrosis in a selective manner, as you can see in the representative images on the left and in the lower right. So the next steps to the clinic are many, but we have proof of concept in these preclinical experiments. We need to optimize dosing and formulation and go through all the necessary steps to reach regulatory filings. So just to summarize, we started with autologous cell therapy in 2004, recognized durable benefits despite cell transients leading to the allogeneic paradigm, identified exosomes as the mediators leading to the idea that we could use intravenous cell delivery in humans. We mined exosome contents to identify specific design factors and then used the recognition of those specific factors to bio-inspire completely new chemical entities, leading to next generation therapeutics and the conclusion of the journey. These are the folks who did much of the work shown, and special appreciation to friends and colleagues below and the sources of funding here. Thank you very much.
Thank you, Dr. Marbán. That was a really great presentation. We're going to jump right into our Q&A session for the last few minutes of the webinar. Okay, first question is, some other RNA drugs have provoked strong immune reactions. Have you seen any evidence of such an effect in your experiments to date?
So we haven't. When we do things like basic metabolic panels and complete blood counts in the experiments so far, the treatment seems innocuous. And I should say that all of the therapeutic benefits that we've seen with injection are actually directionally opposite. We're attenuating in immune and inflammatory reactions, if anything, with this novel agent, TY1.
Okay, great. Our next question says, have you checked the composition of the exosomes in order to determine what is causing the beneficial effect?
Well, I think I answered that in the last 10 slides of the talk. Basically, the RNA content we focused on because light solubilization of the exosomes, enough not to denature the proteins, but enough to degrade the RNA, does away with about 80% of the therapeutic benefit of the EVs when injected. And so that led us to focus on the RNA fraction, and then the RNA fraction led us to focus initially on mirrors, but then eventually on yRNAs and other families, and now has led to the synthesis of these new non-coding compounds.
Okay, perfect. Let's squeeze in one last question. Your new drug, TY1, was bio-inspired by yRNA found in the extracellular vesicles. Can the same paradigm be used to create drugs from other RNA families?
Absolutely. You remember in that pie chart of non-coding RNAs within CDC EVs, we saw large representations of other families, and we have successfully used the paradigm in the lab to create novel piRNA-inspired drugs, and we're in the process of doing so for tRNA fragments that are bioactive as well.
Okay, great response. Well, we have reached the hour, so I do want to say thank you very much for sharing your fantastic insights today, and it was a pleasure to have you with us.
Thank you. Thank you, everybody.
Thanks. All right, and a big thanks also to the audience for participating. In closing, we hope you enjoyed this Inside Scientific webinar, produced in partnership with the American Physiological Society and the European Council for Cardiovascular Research, and we look forward to having you with us again soon.